Well, hello everybody. My name is Andrew MacKenzie. I am in my second to last semester as a master's student at West Virginia University. And today I'm going to talk to you about the avian response to wetland restoration in Preston County, West Virginia. A little bit about me first, just so you guys know who I am. Like I said, my name is Andrew MacKenzie. I have been in Morgantown since 2016. I received my bachelor's in wildlife and fisheries resources with minors in conservation ecology, and then as well as agriculture and natural resources law. I'm pursuing my master's under Dr. Jim Anderson, and I have three proposed chapters to my thesis. The chapter that I'm going to talk to you a little bit today about is pre-active and post-assessments of wetland restoration on biodiversity. So the introduction, my study site is located at the Reedsville Farm in Preston County, West Virginia. Through the Reedsville Farm, there is a place called Ruby Run. Ruby Run is a first order stream, which means it's very narrow and very shallow. And the stream itself is about 1.62 kilometers long, and about 65% of that stretch is within the conservation easement boundary. Ruby Run and the adjacent wetlands were mitigated to offset impacts from a dam renovation for the Arthurdale and Reedsville drinking water. Um, and our pre-restoration surveys occurred from February 2017 through May 2021. The restoration started in June of 2021, and our active restoration surveys ended in May of 2022. And then we continued doing post-restoration surveys from June 2022 through September 2023. On the right-hand side is a picture of the wetlands at the Reedsville Farm. And then this is an overhead view of Ruby Run prior to the restoration. On the left-hand side, that's going to be your upstream end. And then on the right-hand side is going to be on your downstream end. If you look on the right-hand side, you're going to see in just a second that the boundary was increased just a little bit to include a little bit more wetland area. And then this is what the Ruby Run stream looks like now, post-restoration. So we had a fence extension, and then as a part of the restoration, we also graded the banks and removed herbaceous vegetation. When grading the banks, we removed all the herbaceous vegetation, and then we went around into the wetlands and looked for invasive species such as multiflora rose to remove. We also had an aquatic habitat installation as well as an erosion control structures set up. So if you take a look on your left-hand side, you'll see these, what look like down trees. These help provide bank stabilization as well as habitat for aquatic insects. And then on the right-hand side, you'll see a lot of rocks. These rocks are to help prevent the wetland from seeping. After all of that occurred, they planted native or seeded with native vegetation. We used cory mesh to put on top of the seeds, and this helps prevent the seeds from dispersing. So on the right-hand side, there's a picture of what the cory mesh looks like, and then on the left-hand side, there's a picture of what the seed mix looked like about 5 months after seeding. And then the white tree tubes that you see there are from woody vegetation that we installed. The woody vegetation installation had 8 different species of woody vegetation. A majority of the trees were planted in tree tubes, as you'll see on the right-hand side with one of the eastern cottonwoods. In the middle, there is a pin oak. This does not have a tree tube because it was in an area that was prone to flooding, and we did not want to put any tree tubes in the floodplain or areas prone to flooding to make sure that they didn't end up down the river and cause a lot of trash and debris. And then on the left-hand side, you'll see me. That's about the size of a majority of the trees we planted. They're only about 18 inches tall once they had been planted into the ground. Then this is one of my favorite pictures. This is when we had finished the woody vegetation installation. You can see that there are tree tubes all the way to the left. And if you look down to the right-hand side, the tree tubes don't stop. We planted about 1,800 individuals of trees. And as a part of my research, I'll be measuring their survival and growth. And then just to help you guys kind of visualize how this transition occurred, on the left-hand side, we have our wetland before pre-restoration or during pre-restoration. So there's cattle degradation, multiflora rows, and a large abundance of song sparrows. As you continue to the right-hand side, you can see that the restoration starts to occur. Then that third panel is kind of showing how the wetland is starting to establish itself. We started seeing increases in abundance and diversity for several taxonomic groups. The right-hand side is a projected outlook of how this wetland potentially could look in 30 to 35 years. I want to thank Julia Spar from Science Visuals for creating this. And then I will get into the birds. So for our objectives, we wanted to quantify year-round species richness and abundance of the birds and use these observations to test the effects of restoration treatments. Our hypothesis is restoration treatments will not significantly increase the diversity or composition of birds when comparing pre-restoration data to active and post-restoration data. The methods that we used, we did one avian point count survey per month, we did one avian survey per month. With those surveys, there was 5 point counts. Each point count would last 10 minutes. While we walked between point counts, we also observed birds on the transect. We had a radius of 50 meters for each point count, and we used both our eyes and our ears to pick up bird observations. So when I did my analysis, I removed birds that were outside of the conservation easement. I wanted to show you guys this picture because on the left-hand side, you can see there's pasture grass, a fence, and then the floodplain and the wetland. On the right-hand side, you have your pasture grass and your cattle as well. So any birds that were not within the conservation easement were removed, as well as any birds that were outside of our 50-meter radius for the point counts. Once we did this, we started comparing the pre-active and post-restoration data. And for our total abundance, it was a little bit alarming because as we continued into each restoration period, our total abundance continued to decrease. So this is the average amount of birds we observed per month. During our pre-restoration, we averaged roughly 24 birds per month. Our active restoration period, about 22 birds per month. And then our post-restoration, about 20.5 birds per month. When I dove into this a little bit more, I looked at the species richness. Our pre-restoration period had 65 different species. Our active restoration had 35 species, and our post-restoration had 63 species. What I find most interesting about this is our post-restoration period has been only 18 months compared to our pre-restoration period, which was about 4 years. So seeing that we only had two less species in such a short time frame compared to the pre-restoration is pretty promising. One of our main drivers of abundance were song sparrows. And so when you see that large decrease in total bird abundance, it's also good to know that the song sparrows are decreasing. The reason I say that is because when we did our restoration, we were not creating or restoring our habitat for song sparrows. We were focused on more wetland obligate birds. So seeing that song sparrows are declining, but yet still at the wetland is very promising. And then on the right-hand side, I included a nest of song sparrow eggs that I found last year within the wetland right on the stream bank. Red-winged blackbirds are also very abundant at this site. During the active restoration, we were averaging nearly 5 red-winged blackbirds per month. And then during our post-restoration time period, we've seen a decrease to about 2.5 birds per month. Some other notable decreases in observations are American goldfinch, our barn swallow, our eastern kingbirds, and our European starlings have decreased in observations per month. The American goldfinch is decreasing only because several of the goldfinches fly over the wetland. When they fly over the wetland, they are not included in part or in the analysis because they are not actively using the wetland at the time. They're just flying over. The barn swallows, I'm not quite sure why there's a decrease. Same with the eastern kingbirds. And then for the European starlings, there's been a slight decrease during the active restoration. However, post-restoration, they seem to be rebounding. More importantly, the notable increases in observations. I'm going to read these alpha codes first just to make sure everybody's on the same page. But we have increases in belted kingfisher, blue jay, bobolink, Carolina wren, field sparrow, gray catbird, uh, grasshopper sparrow, mourning dove, northern flicker, red-bellied woodpecker, yellow warbler, and then yellow rumped warbler. Several of these species are dependent on wetlands, or sorry, several of these species are dependent on wetlands, which is a good thing for us because we were managing to increase wetland dependent birds. And then for species composition, I'm going to try not to lose you guys, but on the left-hand side, I have put birds that were only observed during pre-restoration. You guys can probably read that list quicker than I can read it off. Some of the birds, such as the Canada goose, the common grackle, and the common raven, are still in the area. Same with the turkey vulture. However, they haven't actively been using the wetland, which is why they are not being considered as observed post-restoration. And then birds only observed during post-restoration include the American woodcock, black-billed cuckoo, northern parula, hooded warbler, tree swallow, yellow-throated warbler, and these are some of the species that we were managing for when we restored the wetland. So it's good to see that they were not in the wetland before, and now they're starting to use the habitat, and hopefully continuing down the road, they'll stay there or stay using the habitat. So for the discussion, conducting pre-active and post-restoration surveys is crucial in helping the restoration efforts. A lot of pre-restoration surveys are 1 or 2 surveys, and that's all they do. It's good to have much more data to have a baseline of the population. conducting the active restoration surveys has been very helpful because we can see how the disturbance has impacted species richness. And then when you have your post-restoration surveys afterwards, you can see what birds are starting to use the wetland and if you need to adapt your management techniques. The restoration efforts have, of course, had mixed impacts on the bird species because we've had several species that are wetland dependent. We have generalists, we have birds of prey. So there's going to be a difference of, or a different impact depending on how the bird's habitat needs. And then, as I mentioned before, active restoration had decreased species richness, and this is most likely due to the large amount of disturbance that occurred in the area. With that, I want to thank everybody very much. I also want to thank my funding agencies that are all listed here. And then I will open it up for questions. My email is on the bottom, and if you want to see any more pictures of the study site, I put my Instagram handle at the bottom so you can look and see kind of how everything progressed. Okay, so Hannah, are the raptors that you detected actually perched within the 50-meter radius of the surveys? Yes, so any of the birds of prey that I detected were either perched on a fence post or they were actively foraging within the wetland. I did have one bird that I did not mention. We had a couple instances or a couple observations of a peregrine falcon using the wetland, and one time I got to watch it actually swoop down, take one of the small mammals, and then it just kind of sat there and ate it. So it was really cool. Can people walk along the wetland and see the restorations? I can't say yes or no to that. Reedsville Farm is a working farm. So I know that you're allowed to go to Reedsville Farm and bird and by the big pond area. The stream that we restored is kind of down the gravel road if you continue. It is fenced off because it's a conservation easement. However, I think that if you were to stay on the gravel road and potentially get permission from the land owners, there shouldn't be an issue. Spencer, thanks for letting me know about the 4-letter codes. I was thinking about that with all the potential name changes for birds, so I appreciate that heads up. All right. Well, if that's all, I appreciate you guys having me, and I will hand it off to Spencer.
Thanks, Andrew.
No problem, buddy.
All right. We're good to go. Everyone can see that?
Yes.
Yes. Okay, awesome. Yeah, my name's Spencer Parkinson. I am in my unofficial final semester. I haven't really taken any classes in a semester, and I actually defended my thesis on Friday, so I'm going to do my darndest to condense this down and keep it from being way too long. So my project was studying if birds are acting as dispersal vectors of Litolanchus crenati subspecies McCanni, which is a nematode that is associated with beech leaf disease. So let's get into it. I'm going to explain a lot of these things later on. So I'm going to give you a quick introduction, talk about plant pathogens and beech leaf disease. I'm going to present to you my research questions. I'm going to go over the methods that I use to address those research questions, and then I'm going to go through my results and my discussions from what we found from our experiments, and I'm going to talk about some directions moving forward. So this is where I would ask people, if I had an audience, who's heard of plant pathogens? We're all familiar with human pathogens, I'm sure, but maybe some people don't know that trees and plants and shrubs also are affected by pathogens and diseases. And the four aspects that we're going to be looking at is the host, which is typically some sort of tree, shrub, agricultural crop, any sort of plant. And then on that host, symptoms are observed. So something that's not quite right with the tree, something that looks abnormal, and it typically is observed on the leaves, as we can see down here in the bottom right in beech leaf disease, the branches, the trunks, as we can see here in chestnut blight and beech bark disease, those cankers around it. And then also in the roots, as we can observe with soybean and potato cyst nematodes. So once we observe these symptoms, we want to figure out what's causing them. And the cause is an organism. It's a fungi, it's a bacteria, it's an insect, it's a nematode, it's a virus, it's an oomocyte. And oftentimes it's more than one of these things. For beech bark disease, it's actually a scale insect that comes and allows for fungi to infect the beech trunk, the trunk. And once we understand the cause, we want to understand the vectors. How are these pathogens and organisms spreading to uninfected areas? So for my project, I focused on beech leaf disease, which is a new emerging plant pathogen that was just described in 2020. We know the host is American beech, or Fagus grandifolia. It also affects other beech trees, such as European beech and Oriental beech. The symptoms are this interveinal chlorosis and puckering of the leaves, as you can see in this picture down on the right. And the cause is a nematode, Little-Ancus crenati McCanni. And for those of you who don't know what a nematode is, which I didn't prior to my project, it is a microscopic roundworm, very different than what we think of as worms, the sectioned worms. But these worms are microscopic, and they're actually the most abundant animal in the entire planet by far. They're super interesting. Look into them. And then the thing that we want to try to understand is, what is the vector? How is beech leaf disease spreading? So it was first observed in Lake County, Ohio in 2012, and we've observed beech leaf disease symptoms mainly on younger sapling trees. This picture on the left is what healthy beech is supposed to look like. Nice, uniform, shiny green color. And then on the right is a beech leaf that has beech leaf disease symptoms, that darkening chlorotic striping in between the veins. And on the left here is a current map of the detection of beech leaf disease by county, by year. Starting in 2012 when it was first observed in Lake County, Ohio. Moving out to these dark blue, moving into 2019 is the dark blue color. Moving into the purple and the pink is 2022, 2021. And then the red is 2023. And then the map, for just comparison, the map on the right-hand side is the current natural range of American beech in the eastern United States. And this map isn't necessarily supposed to be a example of spread. It's actually a map of detection of beech leaf disease. Could have been present in these counties for a long time, but we are now just detecting it there. And something interesting to note about this map is we have a lot of American beech in West Virginia and Kentucky and Tennessee, but for some reason in our map, it doesn't seem to go much further south than Maryland. And so we want to know how it's getting around. How is a microscopic roundworm moving from Ohio to Maine to Maryland? And it sure as heck isn't crawling there. So our theory is birds. Birds are known to be dispersals of a myriad of other things, such as seeds. Birds, over 33% of birds disperse seeds in some manner. Mallards can ingest invasive carp eggs and excrete viable carp eggs. Birds can even eat live animals such as brine shrimp and successfully excrete live brine shrimp in uninfected pools. Chestnut blight is a fungus that has been found on the outside on the feathers of birds. And many common birds such as starlings, pigeons, cowbirds, thrushes are able to consume cyst nematodes and pass the cyst nematodes. And these cyst nematodes contain hundreds of eggs. So they're mature female nematodes, which inside have hundreds of eggs. So our research question, our big question is, are birds vectors of LCM, which is that nematode? And the two lenses we looked through were endozookery, which is just a really fancy word for internal consumption, successful digestion and excretion of a viable organism, or ectozookery, fancy word for external, attachment to feathers, beak, feet, and then eventually shedding that organism and introducing it to a new area. And so the study sites and the timing that we chose for this project is we went to the Allegheny National Forest, which is in northwestern Pennsylvania, and then we went to the Holden Arboretum in northeastern Ohio. And the Holden Arboretum is actually in the same county where beech leaf disease symptoms were first observed in 2012. And we chose these sites because they have extremely high levels of beech leaf disease. Pretty much the entirety of both of these areas are completely covered with infected beech. And we did our sampling during the fall and winters of 2021 through 2023. And the reason we did this is American beech keeps its buds throughout the winter. And the nematodes actually migrate into these buds to overwinter. And as food resources become more scarce for birds during the winter time, they might be utilizing these beech buds, which contain hundreds of nematodes inside them, as a food source. So a broad overview of the methods I used is we established these bird feeders in beech leaf disease areas. We set up game cameras to, one, make sure birds are even coming to our bird feeders, and to two, see what kind of birds we might expect at the bird feeders. We then set up mist nets around the bird feeders. We proceeded to catch birds, and we collected biological samples. And then we used molecular and microscopy methods to look for LCM DNA in those samples we collected from the birds. And we visualized them using PCR and gel electrophoresis. But all we need to know is that we looked and detected for LCM, which is the nematode, in the bird samples. So we use molecular and microscopy methods to detect LCM. Molecular is, like I said, we extracted DNA. We used PCR. The perks of using molecular methods is very quick. You can process a lot of samples in a short amount of time, but you have no way of observing if that DNA came from a viable organism. So we had no way of knowing that. Even though we could detect LCM DNA, we did not know if those nematodes were viable or alive at the time that we extracted the DNA. And microscopy, the perks of microscopy is that you are able to observe viable nematodes. If you are under a microscope and you see the nematode moving around, or you can even observe females with eggs inside of her, which is one perk of working with nematodes, is that they're translucent, so you can actually see all their organelles. But the caveat of microscopy is it's extraordinarily time consuming. So we did use both to look for LCM in this project. And after summing up 2.5 years of freezing out in the woods, catching birds at these bird feeders, here's just a general broad sweeping results. Is we caught 156 birds of 19 different species. And from those birds, we collected 219 biological samples. So for a lot of the birds, we took both an ectozookery and an endozookery sample. So that's why we have more biological samples than we have birds. And then from those 219 samples, we detected LCM DNA in 21 of those samples. And this graph to the left is a species distribution. And I'll just go over the top 5. We had 47 black-capped chickadees. We had 28 tufted titmice, or titmouse. We had 27 dark-eyed juncos. We had 18 American goldfinch and 10 white-breasted nuthatch. And so what was really fascinating is that our methods worked. This is the first time anything's been done like this for foiler plant parasitic nematode, looking for them in avian samples. So it was really exciting that we were actually able to show that we can detect LCM DNA from avian samples. It might not be such a good thing for the beech trees and for the spread of beech leaf disease. It would have been better if we weren't able to find it and could say that birds weren't spreading beech leaf disease, because we don't want to spread beech leaf disease. But we did find that our methods worked. And kind of one of the caveats of our experimental design is that we knew that our species composition was skewed because we were catching birds strictly at feeders. So we weren't getting necessarily a whole snapshot of the forest systems we were in. And here is just a graph or a table of our results by experiment. So for the endozookery, we had 2 experiments. We had one experiment where we kept birds in paper bags for 15 minutes to scare the crap out of them. And then we had another experiment where we kept the birds in cages for 3 to 4 hours. So we had 90 birds that we kept in bags, and from those we had 4 positives. And from the birds we kept in cages, we had 59 samples, and 10 of those came back positive. And then for ectozookery, which again was the external, we dusted for ectoparasites. So we took an ectoparasite insecticide and brushed it through the bird's feathers to kill the ectoparasites and test if we could see LCM DNA in the ectoparasites. We had 55 samples and we had 6 positives. And then we just took pure feather samples. The feather sampling occurred before any of these other steps, before looking for ectoparasites or putting birds in bags or cages. And for this, we collected 3 to 5 from the breast, 1 to 2 from the crown, and 1 to 2 feathers from the vent. And again, for all of these methods, we both looked at microscopy and we used those molecular methods to see if we could find LCM DNA, and we did. The only thing that we were not able to confirm was that the nematodes were viable or motile. So while we were able to observe nematodes in fecal matter and in ectoparasites and in feather samples, we never observed them actually moving, and we could not confirm or say for sure that they were viable and able to continue spreading beech leaf disease. But something that was very interesting from this project is that it seems like ectozookery, or the external attachment and carrying of organisms, was more likely than endozookery. So even though our endozookery had higher percent positives, we did a lot of experimental control to get those positives. We were feeding the birds in cages infected beech buds from beech leaf disease trees. We were actually at one point just feeding them straight nematodes into their mouths and collecting their fecal samples. So the fact that we only had like a 16.9% positive for the caged birds is really surprising. It should have been much, much higher, theoretically 100%, because we knew that these birds were consuming nematodes because we were feeding them to them. And then for ectoparasites and ectozookery, we did no experimental control. This was all purely picked up in the environment. We weren't putting nematodes on the external parts of the bird. So for this, it seemed like ectozookery was more likely, and ectozookery is also more likely to be responsible for long distance dispersal, because there's no time limit on how long an organism can be attached to the outside of a bird on their feathers or on their feet. Whereas endozookery, there's only so much time before digestion occurs in a bird. So there's only a time frame from when the nematode would be ingested to excreted, whereas it could attach to the outside of a bird indefinitely. So we laid the groundwork for a lot of future studies. This whole project is very in its infantile stage. So we explored some methods and decided that there's a lot to be looked at with birds still. We barely scratched the surface, unfortunately. And so some future directions that we need to look at is we want to understand viability over detection. We showed we could detect the DNA in the samples that we collected from birds, but we need to know if those nematodes are viable and able to continue spreading beech leaf disease. And then for endozookery, I think it's really valuable to explore beech nuts. No one has looked to see if the nematode is in beech nuts, and beech nuts are a super important food source for over 30 species of birds and mammals. Ruffed grouse, blue jays, tons of birds eat beech nuts. And then also caterpillars. Caterpillars feeding on beech leaves that have beech leaf disease could be eaten by a bird, and then the caterpillar could have the nematode inside of it, and that could help successfully pass the nematode through the bird's system and sort of act as a protective barrier around the nematode as it's going through the digestive system. And then for ectozookery, one of the caveats of our experimental design is it was intended to kill ectoparasites and kill the nematodes. So obviously we couldn't observe viability when our methods killed them. So washing has proven to be a very effective way of looking for other forest pathogens. So that's washing the feet and washing the feathers in order to collect ectoparasites and nematodes, and then you can actually observe motility and have live viable nematodes to work with. And that just brings us to the final thing is, why do we do this? Why did I spend almost 3 years freezing out in the woods all winter, 6 in the morning? And it's because beech is a super important tree. It's a food and habitat resource for over 30 species of birds and mammals. It's a really important to the forest structure. It's the dominant hard-masting. It's the only hard-masting tree in northern maple hardwoods. And American beech has just had such a hard go at it already. It's been hit by beech bark disease, which came through and killed so many mature beech trees. And in the wake of these mature beech trees dying from beech bark disease, they have regenerating young trees from the root systems. And unfortunately, that's where beech leaf disease loves to run through. So it just seems like beech can't catch a break. It's already affected by a bunch of aphids, a mining weevil, a sooty mold. And if we don't want to see beech go down a similar road that chestnut went down with chestnut blight, it's important to figure out how this is spreading and how we can make predictive models and implement land management to protect uninfected beech stands. So it's an ecological important tree. It's also just a really pretty tree. It's used in a lot of parks. You see people carve their initials into it. Don't do that. But it's got that really nice smooth bark, really big, great canopy. So we need to do as much as we can to try to honestly save this tree. And if we can't figure something out, it could maybe not in our lifetime, but could go pretty much extant from the eastern landscape. And with that, I would love to take any questions. Um, yeah, any idea why it doesn't go south of Maryland? I had originally thought it was just kind of a lack of surveying effort. People maybe weren't looking for it down there, but I was proven wrong at a conference. Um, we're kind of thinking that it's something to do with environmental factors. They're much more prevalent up where they have colder winters. So it could be that the life cycle of this nematode doesn't succeed as well in lower um, the lower states because nematodes have their way their life cycle works is degree days. So a day is based on what temperature it is. So they need to be between a certain threshold. So a theory that I've explored is that in the South, they have more degree days in a row, which makes these nematodes go through their life cycle faster. But that's just no real reason or idea why at this point. They were not. Hannah says, "How willing were the caged birds to eat the infected beech buds and nematodes?" They really did not like us putting beech buds into their beaks. We fed them 3 to 4 every 45 minutes, making sure that at least one of them actually went down because they would oftentimes spit them back up. Um, so they did not like that, and that's one of the reasons we switched to feeding them these beech buds to extracting the nematodes out of the beech buds and making a solution of water and nematodes, and then we would pipette that into the beaks of the birds, and they were more willing to ingest that because that's just nutrient rich. Each sample had 100 nematodes in it, and we did that every 45 minutes. Is there a proposed treatment? I'm not quite sure what that means. Proposed treatment for infected trees, is that the question? Okay. Yeah, they're exploring some things that are used. The most successful treatment that they found is actually what they use to treat turf grass right now, and it's called phosphate 30. And what it does, it just increases the phosphorus uptake of the trees, and that's thought to thicken the cell walls of the leaves because that's what the nematodes are feeding on is the cell walls of the leaves, causing causing irregular cell division and cell layers. So they've had some success with this phosphate 30. The only issues is that it's extraordinarily expensive, extremely time consuming, extremely targeted. So it's most likely going to be used in like arboretums in city parks because it's just it's too intensive to go out and try to save stands and like national forests and stuff like that. Yeah, thank you. Thank you everyone for listening. I hope I didn't lose too many people with nematodes and plant pathogens and all that stuff that's not birds. I lost myself doing this research, so